School of Engineer, in short CIE, and we are San Francisco Bay Area chapter. So this is a organization have a long history, so it's almost 100 years old now. So 2 years later, we will celebrate our 100-year anniversary. So let's introduce the speaker. Dr. Sunil is the Director of Science at Affymetrix. He used to work for the Siemens, have many years experience in the Diagnostics industry. He's a PhD from the State University of New York, and his postdoc down in the Washington University. So let's welcome Dr. Sunil.
Thank you, Simon, for the kind introduction, and thank you all for coming. Thank you as well for inviting me, Simon, and giving me this opportunity to present some of our technologies. It's a great honor and pleasure to be here. Can you guys hear me in the back over there? Yeah. Excellent. Okay. So I'll try to stand over here. All right. So as I was saying, thank you again for coming, and thank you, Simon. It's a great honor and pleasure to be here, to be able to present in front of some of the brightest minds in the Silicon Valley. What I want to do in the next 40 minutes or so is give you guys an idea of some of the Epimetrics technology. I'll give you an introduction to the Epimetrics technology, microarray, which is what it's called, and how we have applied it in terms of prenatal diagnostics, cancer diagnostics, and in agrigenomics. And then I will digress and move away from the Affy technology and talk about what else is happening in the field of genomics. There's a lot that's happening in the world around us, and I'd like to just give you guys a little flavor of that, followed by talking a little bit about precision medicine, companion diagnostics, and how all of this is shaping up the healthcare for tomorrow. So the title of my talk is DNA Microarrays, Current Technology and Clinical Applications. All right, a little bit about Affymetrix technology. Affymetrix, where we are not too far from here, just off Central Expressway, a few miles down, headquartered in Santa Clara, 1,100 employees worldwide, incorporated in 1993. We have 11 worldwide offices with more than 500 patents and more than 94,000 publications in which Affymetrix has been cited around the world, and we serve everything from biotech diagnostics to pharmaceutical companies and number of leading academic institutes as well. Bio 101, I apologize, we have some very bright PhDs over here from Caltech, Stanford, Johns Hopkins. Apologize, this is just meant for some of the engineers. Just to remind you guys, Molecular Biology 101, that we are all our cells in our body have DNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid, and proteins are proteins. These are the key molecules inside a cell. And DNA over here is basically a double-stranded molecule. This is the sense strand. This is the anti-sense strand. They're complementary to each other. And we all, on most of our cells, inside our nucleus, we have 3 billion base pairs of DNA. And this is our genetic code. And from DNA, you can make RNA through the process of transcription. And from RNA, you can make proteins. Okay? And this process, from one molecule of DNA, you can make many molecules of RNA. From that, you can make many molecules of protein. Okay, so this is a process for amplifying the signal and also to regulate the signal. All right, DNA, just one more slide on basics of DNA. For some of us who are not familiar with this, DNA is a double helical structure. It is made up of nucleotides A, G, C, T. So unlike the software world with code of 0 and 1, we have a 4-letter code over here of ACGT that determines who we are, what we are, and what diseases we're going to get in our lifetime. All right, and then the ACGT is based on we have bases and we have sugar. Okay, so the bases can be either purine or pyrimidine, and A and G are your purine bases, nucleotides, and C and T are your pyrimidine. And then the sugar can be, it's a 5-carbon sugar, it's a pentose sugar, and it can be the ribose sugar, which is part of the RNA, or it can be a deoxyribose sugar, which is part of the DNA. That's why it's called deoxyribonucleic acid and ribonucleic acid, DNA and RNA. One last reminder, the A always base pairs with T, and G always base pairs with C on the complementary strands. So you will have these A-T base pairing and G and C base pairing. So there's 2 hydrogen bonds between A and T that hold them together, and then there's 3 hydrogen bonds between G and C that hold them together. And that's how the 2 strand remains together, and it's wounded. It's a helical structure with these hydrogen bonding and sugars all facing outside, sugar and phosphate. So just keep this in mind. This is just molecular biology 101. All right, so what are microarrays? Microarray is basically a device that tests for the presence or absence of specific DNA or RNA in a sample. And really, when this was invented, it really revolutionized the way in which biology was done. Previously, before this technology was invented, we were looking at single genes and their expression or RNA from the genes at single molecule level, at single gene level. But by this technology, you could look at it at a very large scale. You can look at all the genes inside in an organism using this, or in humans or in other organisms using this technology. So the history about this technology, microarray technology, it was founded by Stephen Fodor, and they invented this. Again, came out of the Stanford corridor in late 1980s. They published a landmark paper in 1991 describing massively parallel synthesis of arrays. And so this over here was the first prototype of the array, just done on a glass slide, and then this is the current array. And I have samples of the array in my bag over here. I'm going to pass it around so that you guys can take a look at it to see how it looks. Here, pass it around. So, and we have some newer generations of those arrays that we can do in 96 and 384 format. Basically, the theory behind this work was that notion, and this was really borrowed from the semiconductor industry, this technology, that you could use semiconductor manufacturing techniques and in combination with combinatorial chemistry to build vast amounts of biological data on a glass slide or a silica chip. And so again, this is borrowed from the semiconductor industry. Basically, this is a silicon chip that some of you guys use in your computers and stuff. And then using the process of photolithography, which is a process of using light and light blocking mass to expose and activate specific patterns on a substrate. So what you're able to do with this is that you're able to put, using this kind of photolithography process, which borrowed from semiconductor industry, you're able to make the DNA oligos on top of this chip. And the way that process works is that you have your wafer over here, and when you shine light on selected portions of this, the portions where light shines, it gets deprotected. It's basically the photolithography process. And then you throw in your nucleotide, your base, your nucleotide T, that will go and bind to the regions that are deprotected, and the others will be still protected. And along with the T comes, it is all when the nucleotide gets added, it's still protected. So in the next round, when you shine light on some other areas, those areas get deprotected, and then you can add a C over there, and so on and so forth. So using this process of protection and deprotection, you can make oligos or strings of DNA nucleotides onto this chip surface. And we use all the processes, similar processes like the semiconductor industry. We have to wear the white bunny suit as well to keep everything clean and neat. Okay, so basically, this is the big wafer, which is cut into these tiny wafers after the DNA is synthesized, and this tiny little portion, which can be anywhere from a centimeter, 1 centimeter by 1.2 centimeter, and each of these, they have a small. I'll show you a better picture in the next slide, but in this 5 micron by 5 micron region, you'll have millions of these DNA oligonucleotides that are synthesized over there. Now each of these, so if this is your entire chip over here, which is your 1 centimeter by 1 or 1.3 centimeter by 1.3 centimeter, and this is your tiny little feature over here of 5 microns by 5 microns. Each of these features has the identical DNA sequence of 25 nucleotides that is made over here, and you have millions of these in each of these features over here. Okay, and again, this is using the same photolithography process of protection and deprotection, and so in terms of manufacturing, because it is so precise, we have this is a very precise process, and we from batch to batch variation is very minimal or non-existent. So once you have a chip like this, so you have different sequences on different regions, so you can cover an entire genome or an entire DNA sequence of an organism. These days you can put an entire 3 billion base pairs that you want to probe in one experiment, in one chip experiment, using this, these kinds of the chip arrays. So how do you do that? This is sort of a very simplified picture of how we do that. What happens is basically, once you have the DNA strand that is synthesized on the array surface, this is called the capture probe. So this is your capture probe that you're synthesized on the surface of the chip. And the other strand is a tube of sample or target that's to be tested. So let's say you want to test, say, my DNA. You will take my DNA, chop it up, and you will label it with a fluorophore that is attached to my sequence. So what happens when you you add my labeled DNA sequence on this target? What happens is targets are labeled with the molecule that binds fluorescent dye, and so when the labeled sequence will go and look for its complementary sequence on the chip, right? So some of these which do not match the sequence perfectly will not bind to the sequence on the chip, whereas there are other 25 or sequences that will, if they find the complementary probe on the chip, they will go and bind to that area on the chip. And then, because they are also attached with the fluorophore, you can detect this, because you see these areas are red as example, which means that this is where the labeled probe has found its complementary region on the chip, and you can detect that. So we have entire instrument and software and all the downstream suite that's built around this arrays, so that we can do this in an automated high throughput fashion, and once this kind of hybridization is done, where the complementary probes have been found, you can go and scan the chip to find where exactly is the signal, and you can also do this quantitatively. And this way you can see differences between, say, normal or a tumor, or differences between two individuals and things like that. So this really allows the biology to do in a high throughput manner, because you can probe the entire genome of organism using just one chip experiment. All right, so shifting gears a little bit, let me talk about how the chips have been used at Epimetrics, and I'll talk about those in 3 different areas. But this slide over here shows the mission of Affymetrix, which is biology for a better world. And imagine if you could translate the deep understanding of biology in laboratory into routine practice in the clinic and in the field for improved human health and wellness. And that's the mission Affymetrix is after. It's biology for better world. This slide just gives you a product snapshot. Traditionally, Affymetrix first started about when it started 20 years back. It was highly in the research market, where it was serving most of the academic institutions. They were doing all research type experiments to see how does a tumor differ, what are the genes that are uniquely expressed in a tumor, and trying to understand the biology. Now we have moved away quite a bit from just the gene expression business, and we have a number of products out there in terms of childhood disorders and arrays, microarrays to detect developmental disabilities, and I'll talk more about that in my subsequent slides. We also have a product in the oncology area where we can detect mutations again at the whole genome level, and we also have a product over here where we can look at RNA molecules, ribonucleic acid molecules at a single cell level using this technology, and we can do that quantitatively. But I will not talk more about this. I'm going to skip that. But we have that technology as well, and we're working on that. So 3 areas are agrigenomics, reproductive health, cancer diagnostics. Let's look at agrigenomics first. So one of the challenge and Epimetrics' mission is to make sure that no one goes hungry. I was like, how are we going to do that? When I first joined, I just joined Epimetrics a month back, and the HR guy tells me one of our missions is to make sure nobody goes hungry. Okay, really? How are we going to do that? Okay, challenge feeding the world. So by 2050, the population of the world is going to surpass 9 billion people, which means we're going to need 33% more food to feed the population around the world. And to feed, so the population will be approximately 9 billion by 2050. And so in 2050, if the population is about 9 billion, 30% more, we're going to require 100% more food. And to do that, to have 100% more food, we'll have to make better products, use technology, and also use better ways to do our breeding experiments and then and generated traits that we're going to select for. So we have to make better crops or better cattle or better cows so that we can get more food for the same. So 70% of this food must come from efficiency and improving technology. And this is some guy named Jeff Simmons from Elanco Animal Health. It's his quote. So how are we going to do that? Agrigenomics. So currently, when some of these breeding experiments happen, it's more like a trial and error where for the farmers use physical traits for, say, corn or cattle. Just a couple of examples here. But using Affymetrix technology for defining variants, and we can use the microarray technology to define at the genetic level which organisms, which cow or which corn is a better breed, and we can monitor the crosses, and rather than just making a random guess and using trial and error method, and that can really help in terms of increased production of say more milk producing cows, more disease resistant crops, and so on and so forth. And this is shown more in the next couple of slides. I've shown some examples. So we have designed genotyping arrays. We are one of the world leaders in this area, and we have designed genotypic arrays for all these organisms shown in this picture, from bovine, chicken, equine, salmon, catfish, wheat, barley, sunflower, all of these. And we have products in all of these areas. We have one of the largest portfolio of genotyping arrays in the agrigenomics area, and it is really helping farmers and people worldwide in terms of doing the right selection. Okay, and here's few examples that around the world that are shown over here. There's companies like Agrigenomics and that are helping the meat and egg production from chicken. Here's an example from a company where the salmon are affected by sea lice, and so you have to give them excessive antibiotics. So if you can find breed varieties of salmon that are resistant to the sea lice by breeding experiments, you don't have to give them that much antibiotics. Similarly, over here, University of Bristol is looking at making better wheat production and better wheat yields. And then there's a company called Lima Grain. It's one of the fourth largest company in terms of all these grains, and the fourth largest grain company in the world. And they are using our arrays as well for a number of different crops to get better crops. And the chip technology that I showed you, and I'm passing it around, that's a single chip out there. What we have done is to screen some of these in a high throughput manner, and to further increase the throughput, what we have done is we have put the chips in this 96 well or 384 well format, where you can have 384 or 96 chips that you can probe simultaneously, or you can probe 96 different varieties at the same time. Okay, what are we doing? What is Epimetrics doing in reproductive health? Just some facts. 13 to 14% of US children have some sort of development disability. And this has been growing up in the last 12 years. So cases of ADHD, attention deficit hyperactivity disorder, so things like autism cases have tripled in the last 12 years. And also the birth defect tops list of causes of death in US CDC report. This is a 2010 report that birth defects such as low birth weight, sudden infant death syndrome are some of the 3 top causes of death in infants. And for some of the development disabilities, what happens is a child is born with one of these disabilities. You take them to the doctor. The doctor doesn't know really. He says, come back in 6 months. The parents also don't know. They're also left in dark. And by the time all of this cycle happens, the child is almost 4 years old. And for all those years, the parents are left in dark and things like that. So the sooner we can detect the problems, the earlier we can intervene and take care of those problems. There's some statistics over here which says that the lifetime cost of autism can exceed about $2 million per person. So if there's a child that's born with or is autistic, on average it's going to cost the healthcare $2 million per person to take care of the child. And what are some of the methods that have been done traditionally? Traditional methods have been just looking at chromosomes, which is a very, and this has been going on for 100 years. Karyotyping, where what they look is they look at your chromosomes in the cell, and then they look for microscopic analysis of the structure of chromosomes, and so but with this technique, it's very, and you cannot see really small levels of deletion. These are some gross changes that you can see at the chromosome level due to this staining method, but it's not very sensitive method. And then there's other methods such as FISH, where you can go in and look at specific regions in the chromosome. By the way, I forgot to mention each cell in our body has 23 pairs of chromosomes, and the 3 billion base pairs of DNA is divided into these 23 chromosomes that are present in every cell. So the bottom line is these methods are there. They've been there for 100 years. They don't work very well. They're not very sensitive. And what we have developed is a cytoscan diagnostic array. This is FDA-approved product. We are a highly regulated industry, and everything that we do for testing patients has to be approved by FDA. And so this Cytoscan DX assay, which is again based on a chip, it's the first FDA-cleared whole genome diagnostic test to aid physicians in identifying underlying genetic causes of development delay, intellectual disability, congenital abnormalities, or dysmorphic features in children. So you can look at all the chromosomes, the entire DNA of a child in one shot experiment, and it is much more sensitive than the existing methods of karyotyping and FISH. And you can even do this so I showed you example over here. This is postnatal, after the child is born, but you can also do this prenatal when the child is in the mother's tummy, and you can look at the chromosomal microanalysis. And this was a recommendation that came out in late 2013 from the American College of OB/GYN on genetics for maternal fetal medicine. And they recommended that when a mother goes for ultrasound, and if you see abnormal ultrasound, you do this microarray analysis using amniocentetic fluid or other. So you can get small number of cells, and you can do this analysis even before the baby is born. Switching gears, let me talk a little bit about cancer diagnostics. So just again some facts. Cancer is the leading cause of death. It'll affect about 7.4 million deaths worldwide due to cancer. It's the second leading cause of death. Heart disease is the number one killer. And particularly in the developed world, such as the United States and other developed countries, there'll be 1 in 3 people who will develop cancer during their lifetime. And not only that, once you get cancer, the lifetime risk, 1 in 9 people will have a risk of redeveloping the cancer again once they have survived the first one. So what are we doing in terms of how are the arrays, microarrays helping cancer patients? We have another product. Again, it's a microarray, the chip-based DNA array-based technology, where you can in 48 hours generate a result from looking at the DNA from a cancer patient, and you can even look at you look at the DNA from the tumor, and sometimes what the pathologist will do is they'll take the tumor tissue and put it in embedded in paraffin. So we can, using this technology, we can go back and look at a archived tissue as well, archived tumor material as old as 20 years old material that we can go back and probe this for what are some of the aberrations or what are some of the changes that are happening in the DNA of the tumor. And this result, we can get the result in 48 hours. We can look at, again, entire genome, 3 billion base pairs of, and this includes about 900 cancer genes, and so on and so forth. So again, this has really revolutionized the way the biology is done. Rather than looking at one gene at a time or one piece of DNA at a time, you're looking at the entire and getting answers from that. What else is happening in the world of cancer diagnostics? I just want to throw one idea out there that Affymetrix is interested in as well, but there's already technologies out there that are being developed by engineers such as you, chemical engineers who work on and mechanical engineers who are working on microfluidics-based solutions to detect these rare cancer cells that are released in blood. So here's a figure. So this in blue over here is shown is a tumor that has grown. It's cancer, solid tumor that has grown bigger, and now it's touching a blood vessel. Once it touches a blood vessel, some of the cells will will penetrate through the blood vessel and go to other sites. So let's say you have breast cancer, but then when the cells get into the blood, they can go and to other locations and go. So then you'll have a cancer and a tumor. It is called the process of metastasis, where it will go to lung or a liver or other places, and that's how the process happens. But these cells, these are called circulating tumor cells because they're circulating in your blood. These are very rare. So just to give you a scale of what this technology needs to detect is that if you take 10 mLs of blood, that 10 mLs of blood has 10 billion or more red blood cells. And there'll be hundreds of millions of white blood cells in that blood. And so you can have anywhere from one or more of the circulating tumor cells. So you need to be able to detect that kind of, you need to have that kind of sensitivity to be able to detect the cell. And people are trying to take this single cell and trying to do molecular analysis, DNA sequencing. You probably heard a lot about DNA sequencing. And this is a technology, this is the first generation technology that came out. This is FDA approved for breast, colorectal, and prostate cancer. This is developed by J&J, Cell Search, Johnson & Johnson, and now they're coming out with a third generation microfluidics-based chip that can do this kind of detection of circulating tumor cells. It's a great example of collaboration between engineers and biologists and doctors to find a solution to this problem and develop sensitive technologies. And a lot of the revolution that has happened in biology, it's happened later than other fields because we needed help from all these engineers and chemists and other technologies to support us and develop these technologies so that we could use this to answer biological questions. In the next 10 to 15 slides, I want to talk about where else is the biology going and what is happening in the field of genomics. I cannot talk about everything that's happening, but some of the areas that are of interest to me, and I think it's worthwhile for you guys to hear as well, is some of these things that I'm going to talk about. I want to introduce you to a concept of single nucleotide polymorphisms, or SNPs as they are called. So SNPs or single nucleotide polymorphisms are a one-letter variation in DNA sequence. SNPs contribute to differences among individuals. The majority have no effect. Others cause subtle differences in countless characteristics like appearance, while some affect the risk for certain diseases. So here's, say, DNA sequence from two individuals, person 1 and person 2. So something like this, in one person's sequence there's a T, in the other person's sequence it's a C. And this occurs at a frequency of more than 1% in the population. And so these are called SNPs. So here's a fact for you. This is Will Chamberlain, 300 pounds, 7 feet 1, very well-known NBA basketball player. He died, but he had 2 NBA titles. And here's Willie Shoemaker. He was a jockey, very well-known jockey. Only 4 feet 11, 105 pounds. He won number of derbies. DNA sequence between these two individuals, 99.9% identical. Only 0.1% difference in the DNA sequence of these individuals. But if you look at 3 billion base pairs, you do 0.1% of 3 billion base pairs. You do the math. So that's what contributes to these differences. That's what makes Willie Shoemaker and Will Chamberlain. Variation in eye color, blue eyes, brown eyes, black eyes. Again, because of these variations in SNPs. Complex traits. Francis Collins, who's now currently the director of NIH, who used to be the director of National Human Genome Research Institute, he said, finding the genes that contribute to the common conditions like Alzheimer's disease, cancer, schizophrenia, diabetes, autism, asthma, it's critically important, but it's also terribly difficult because it's a complex disease. Every gene has very small effect in this case, like for some of these disorders. And environment also plays a huge part. So 50% is genetic, 50% is environment. How do you tease out all of this to really lay your hands on what leads to Alzheimer's or schizophrenia or autism? In 2003, and this was effort that was happening for many years, The genome community, or some of the Francis Collins and others, they declared the Human Genome Project complete. This was the first time that the DNA of a human was sequenced fully. 2003, it cost about $2.7 billion to sequence 3 billion base pairs. At that time, the technology was at that. This was only 2003. So, and not only did they sequence the human, but they also sequenced all these other organisms and said, how similar or different are these organisms from humans? So if you look at the number of genes, or the number of base pairs, or number of human equivalents for yeast, fruit, flower, mouse, yeast only has, this is your baker's yeast, used for making bread. It has 6,000 genes, 12 million base pairs, and it has 31% equivalent genes in humans. Whereas fruit fly, your Drosophila, it has approximately 14,000 genes, 165 million nucleotides, and 50% of the equivalent genes are present in the human genome. In mouse, the mouse genome is about the same size as the human genome, and it has almost all of the mouse genes are present in the humans. And interestingly, chimpanzee and humans are 98.8% identical. So between two humans, the identity is 99.9%, but our nearest relatives, chimpanzee, 98.8%. And all of this was possible because of the sequencing efforts and on the Human Genome Project. It has also had a heavy impact in a number of different areas. Because of these genomic studies, we can look at diseases in a new way. It has helped in drug discovery, identify new targets, new drugs. It has helped in diagnostics, helping developing specific diagnostic markers, and also in terms of treatment. In terms of treatment, what the current medication is, that you go to a doctor, what does a doctor do? The doctor measures your height, measures your weight, measures your blood pressure, and prescribes your medicine based on that. So everybody gets the same medicine. Maybe the dose may vary a little bit. But this overdosing, underdosing, or misdosing based on just using height and weight and body weight is costing about $100 billion a year. And this one-size-fits-all therapy ignores the incredible genetic diversity of humans. And also, bad reactions to medicines. Doctor asks you, what are you allergic to? It's just based on your information. And if you don't know, you could have a hypersensitive reaction to some medicine. And this kills about 100,000 Americans a year based on allergic reaction. And again, different people are different. I told you, based on SNPs, people are different. And this is also, SNPs also help you predict individual drug response, how different people respond to different medications. Here's an example how the chip technology has been used for warfarin dosing. Warfarin is a blood thinner. People who have had strokes need to be on blood thinner for the rest of their life. How do you define the right dose? Again, the doctor is, you have to, takes about a month or 6 weeks for the doctor, and they have to keep adjusting the dose, and you can overdose or underdose. And it's very critical that you have the right amount of blood thinner, because if you give too much, you're at risk of bleeding to death. Or if you get too little, you can have another blood clot and you can have another stroke. So so the chips, we have a cytochrome P450 chip. This is an enzyme that determines how your drug is metabolized, whether you're fast metabolizer or slow metabolizer. And that combined with these other genes, now you can do a genetic test to determine what is going to be your right dose for warfarin. Same thing for cancer. 75% of the cancer drugs are ineffective. The doctor uses, different medications, but most of them don't work, and you end up losing your hair and so on and so forth. So I want to introduce you to a concept of precision medicine, and this was also brought up by our President Obama. and he has done this initiative where you can give the right drug to the right patient. You can determine a priori which patients are going to respond to the medicine and which patients are not going to respond to the medicine. And what that leads to is if you do the test earlier, and then there's a couple of examples over here in the oncology area where there's a drug specifically targeted for non-smokers who get lung cancer and in the Asian population. And this is just about 3 to 5% of the population. But if you have that mutation or if you have that aberration, you can get this drug. And what this leads to is in a traditional drug development process, it takes about $1 billion and 10 years to develop a blockbuster drug such as Lipitor or other drugs that are out there. But this way, when you have a targeted drug, a drug targeted to specific mutation, you can test the population, you have a higher success rate of the drug making it to the market and not failing in the hands in one of the trials. And here's a couple of examples over here where instead of traditional 10 years, it took about 3 to 4 or 5 years for the drug to get out, and it is so basically it is you're going from one size fits all to going to a niche model where you've targeted therapy and where you've stratified the patient population of who's going to respond and who's not going to respond, and then develop the medicines that way. So currently, all the pharmaceutical pipelines require you to test your clinical trial population before you do the drug development. There's DNA sequencing technologies since the days of Human Genome Project, where it cost $3 billion, $2.7 billion to sequence the DNA. There's companies out there such as Illumina, Thermo Fisher Life Technologies. Then there's Genia that recently got bought by Roche. And then there's BGI, Beijing Genomic Institute, which is one of the largest sequencing powerhouse in the world. That they also bought Complete Genomics over here. And these guys, and Foundation Medicine, which is offering sequencing capabilities to cancer patients and get sequencing the tumor DNA and offering sequencing services based on some of these technologies that I showed you. And just like the cost of chip went down when the first chip came out 30, 40 years back, it was very expensive. Now the cost of chip has gone down just based on Moore's law. And similar, the cost of sequencing per base and per genome has been constantly going down since the early days of 2001. And it is almost down to $1,000 with the technology improvement. But the goal is to raise to a $100 genome to be able to sequence. It won't be too far in the future when every child and their genome will be sequenced. Already, some of these sequencing technologies are being used in cancer diagnostics, where you can take the tumor of a patient who is not responding to any of the medicines, current cancer medicines, and you can do some of these genomic technologies and see, based on this analysis, that if they can, there's other drugs out there that you can use to treat the cancer patients. Here's a very touching example of a child, and this is not in cancer, but this is in this child, and this study came out of the Medical College of Wisconsin. It's a 6-year-old child, Nick. He had severe inflammatory bowel disease, and he had 100+ surgeries, couldn't take any food, and the doctors couldn't diagnose what was going on. Okay, but then what they did was they sequenced the genome of this organism. This boy Nick, and they found out that he had disorder in this gene. They gave him stem cell transplantation, and the child is doing very well. So there's numerous examples like this of using genome sequencing technologies to be able to treat and use this in clinical application. And I think I'll skip the last slide, and thank you very much.
Thank you, Dr. Sunil. Actually, the first time I got his slide, it's more than 60 slides, right?
Yes.
He's already tried to cut it. I hope I wish I have, the 2
I'm sorry.
We have a lot of things want to share with everyone. Anyone want to have a quick question? 1, 2. Just 1 week ago, President Obama announced a very big project, 250 million US dollar, want to sequence a million American people. What the science is expecting can be found from this project?
So that was one of the slides that I removed in and moved it to the back of slides. So here, so this is what Obama wants, and I told you guys introduced you to the concept of SNPs, right? How different individuals differ. So by using, I don't know how many individuals did he say he's going to do, a million? Okay, so what basically you're looking for is to getting all the genetic information from all these individuals, correlating that with their drug history, their family history, what diseases they have, their lifestyle and everything, and looking at the DNA level and looking at what are the similarities and what are differences in these subset of individuals, and seeing which one of these changes contribute to this one, or which one of these changes contribute to this disease, or which one of this. So that's the plan. Okay, so in the slide over here, I've shown over here. So your health is linked to the health of others through genetics. Sharing your health data helps you. So here's all these individuals, and that's you. So we're building an interactive community, sharing knowledge and data at scale, and that's basically the goal of the precision medicine paradigm.
Thank you. Let's thank Dr. Sunil one more time.
Thank you very much.
CIU, we want to present a small token to you.
Oh, thank you very much. It's very nice.